Students, further we will study about the development of ovule. The ovule develop from the inner surface of the ovary, which is lined with a special ring of tissue, the placenta. The ovule primordia appear as a small mass of homogeneous tissue on the surface of the placenta. It grows rapidly and assumes a conical structure with a rounded tip. This mound-like structure is the future nucellus. It increases in size by the repeated division and project out. As the development continues, the integument primordia originate close to the base of the nucellus tissue and grow upward, forming the integument. In bitegmic ovule, the inner integument grow faster than the outer integument. Although the integuments are initiated after the nucellus, they develop faster and completely cover the nucellus except in the micropylar region. Megasporogenesis. Megasporogenesis, the various steps leading to the formation of the megaspore, is called megasporogenesis. Formation of megasporogenesis takes place as follows. A single cell in the hypodermal region of the nucellus functions as an archisporial initial. The cell is differentiated during the early stage of the development. The archisporial initial is recognized by its large size, prominent nucleus, and dense cytoplasm. Further development of archisporial initial depends upon the type of the ovule. First type is the tenunucellate type. In this type, the archisporial cell directly function as the megaspore mother cell, so that the sporogenous cell is hypodermal in origin in nature and is surrounded by single layer nucellus, as in case of Ellipteria aculis. Crassinucleate type. In this crassinucleate type of ovule, the archisporial cell divides by the periclinal division, forming an outer primary parietal cell and an inner sporogenous cell. The parietal cell may remain undivided and undergoes periclinal and anticlinal division, forming several parietal cells, thereby pushing the sporogenous cell deep into the nucellar tissue. The sporogenous cell is thus subhypodermal in nature. Example in Myriophyllum intermediatum. Pseudocrassinucellate type. In pseudocrassinucellate type, the the sporogenous cell becomes subhypodermal due to the division in the nucellar epidermis. Example in Nigella damascena. Davis in 1966 reported the tenunucellate ovule from 105 family, crassinucellate ovule from 179 families, and pseudocrassinucellate from 11 family. Single cell archisporial initial is the most common, but in the Chromometaceae and Poenaceae it consists of a group of cells, where in sedum the archisporium is multicellular in nature. Second, the primary sporogenous tissues. The primary sporogenous cells or functions directly as a megaspore mother cell or the megasporocyte, but in member of sympathy, the primary archisporial cell function as a megasporocyte. In either case, the megaspore mother cell divide by the reduction division, forming a dyad, which again divide to form a linear tetrad. In some form, as in Rumex and Rheum, the T-shaped tetrad have been reported, whereas isobilateral and tetrahedral tetrad have been reported from Crassulaceae and Hydrocratiaceae respectively. The four haploid cells of the tetrad are known as the megaspore. Of the 4 megaspore, the megaspore lying toward the chalazal end remain functional, whereas the remaining 3 megaspore degenerate. However, which and in what form the megaspore remain function to the embryo sac shows variation in the angiosperm. The megaspore is the first cell of the megasporophyte, that is the female gametophyte. Thank you.